Welcome to Kalank Igniting Curious Mind. Sexual reproduction in flowering plants. Are we not lucky that the plant reproduces sexually? The myriads of flower that we enjoy gracing it, the scents, the perfume that we are sold over, the rich color that attract us, all these are aid of sexual reproductions. Flower do not exist not only for us but to be used for our own selfishness. The flowering plants show a sexual reproduction. A look at the diversity of the structures of inflorescence, flowers and the floral plants shows an amazing range of adaptation to ensure formation of end products of the sexual reproduction, the fruits and the seeds. In this chapter, let us understand the morphology, structure and the process of sexual reproductions in the flowering plants. Flower, a fascinating organ of angiosperm. Human beings have an intimate relationship with the flower since immemorial. Flowers are objects of aesthetic, ornamental, social, religious, cultural value. They have always been used as a symbol for conveying important human feelings such as love, affection, happiness, grief, mourning. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out names of the 5 more flowers that are used in social, cultural celebration in your family. Have you ever heard of floriculture? What does it refers to? To biologists, flowers are morphological and embryological marvel. And the sites of sexual reproductions. In the early classes, you have read about various parts of a flowers. Will help you to recollect the parts of typical flower. Can you name the 2 parts of a flower in which the 2 most important units of sexual reproductions that develop? The pre-fertilization, structure and the events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has to be taken. Several flower hormonal and structural changes have initiated that leads to a differentiations and further development of floral primordium. Inflorescences are formed which bears a floral bud and then the flower. In the flower, the male and the female reproductive structure, androecium and gynoecium, differentiates and develop. You would recollect the androecium consists of whorls of stamen representing male reproductive organ. Gynoecium represent female reproductive organ. Stamens, the microsporangium and the pollen grains. In the figure, it shows that the 2 parts, typical stamen, a long and slender stalk called filament, and a terminal that generally bilobe structure called as anther. The proximal end of the filament is attached to thalamus or the petals of the flower. The number and the length of the stamens variable in the flowers are different species. If you ever to collect a stamen, each of 1 to 10 flower, each from different species, and arrange them in a slide, or you would be able to appreciate the large variations in size and in the nature. A careful observation of each stamen under the dissector microscope making neat diagram would elucidate the range of shapes and attachment of anther in different flower. A bilobe, typical angiosperm anther is bilobed, with each lobe having 2 theca, dithecous. Often our longitudinal grooves run lengthwise separating the theca. Let us now understand various types of tissue and their organizations in the transverse section of an anther. A bilobe nature of an anther is very distinct in transverse section of an anther. Anther is 4-sided tetragonal structure consisting of 4 microsporangia located at the corner, 2 in each lobe. A microsporangia develop further and becomes pollen sac. They extend longitudinally all through the length of an anther and are packed with the pollen grains. The structure of microsporangia. In a transverse section, a typical microsporangia appear near circular in outline. It generally surrounded by 4 layers: the epidermis, endothelium, middle layer, tapetum. The outer 3 layers perform the function of protections, helps in dehiscence of anther to release the pollen. The innermost layer is a tapetum. It nourishes the developing pollen grain. Cells of tapetum possess a dense cytoplasm, generally have more than 1 nucleus. Can you think of how tapetum cells would become binucleated? When the anther is young, the group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangia, microsporogenesis. As the anther develops, cells of the microsporogenous tissues undergo meiotic divisions to form microspore tetrad. Would you be ploidy? What would be the ploidy of the cells of the tetrad? As each cells of sporogenous tissue is capable of giving rise to a microspore tetrad, each of them is a potential pollen, microspore mother cells. The process of formation of microspore mother cells, PMC, through meiosis is called as Megasporogenesis. The microspore they are formed or arranged in a clusters of four. The microspore tetrad has the anther mature, dehydrates, the microspore dissociate from each other, develops into pollen grains. Inside each of the microsporangium, several thousands of microspores or the pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grain represents the male gametophyte. If you touch the open anther of a hibiscus or the flower, that would find deposition of yellowish powdery pollen grains on your finger. Sprinkle them on the top of a grain on the drop of water taken on the glass slides. Observe under the microscope. You would realize the amazing variety of architecture, size, shape, color, and design seen on the pollen grain. From different species, the pollen grains are generally spherical in measures, 25 to 50 micrometer in diameter. It is a prominent two-layered walls. The hard outer layer called exine is made up of a sporopollenin, which is one of the most resistant organic known. It can withstand high temperature, strong acids, and alkali. No enzymes or degrade sporopollenin is so far known. The pollen grain exine is a prominent aperture for the germ pores, where the sporopollen is absent. The pollen grains are well preserved, have fossil because of presence of sporopollenin. The exine exhibits a fascinating array of pattern designs. Why do you think the exine should be hard? What is the function of the germ pore? The inner wall of the pollen grain is not called as intine. It is thin, continuous layer made up of cellulose pectin. The cytoplasm of the pollen grain is continuous, surrounded by the plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. The vegetative cell is bigger, has abundant food reserve, a large irregular shaped nucleus. The generative cell is small, floats in the cytoplasm of a vegetative cells. It is a spindle shape with a dense cytoplasm and the nucleus. It's over 60% of angiosperm pollen grains are shed in two cell stage. In the remaining two species, the generative cells divide mitotically to give rise to two male gametes. Therefore, the pollen grains are shed in three cell stage. The pollen grains are of many species cause a severe allergy, bronchial affiliations in some of the plants, leading to a chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that the Parthenium or the carrot grass has came into India as a contaminant with the imported wheat, has become a ubiquitous in occurrence and causes pollen allergies. Pollen grains are rich in nutrition source. It has become a famous in the recent years to use pollen tablets for supplements. In the Western countries, a large numbers of pollen product in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of an athlete and a racehorse. When once pollen grains have led to land on the stigma, have become lose viability if they have to bring back the fertilizations. How long do you think the pollen grains retain the viability? Period for which a pollen grain remains viable is highly variable and to some extent depends on the prevailing temperature, humidity. In some cereals such as rice and wheat, pollen grains loses viability soon within 30 minutes of its release. In some members of Rosaceae, Leguminosae, Solanaceae, they have maintained viability for months. You may have heard of the shortening semen sperm of many animals including human for artificial inseminations. It's possible to store pollen grains for a large number of species for years in the liquid nitrogen, minus 96 degrees Celsius. Such store of pollen can be used as a pollen bank similar to the seed bank in the crop breeding program. The pistil, megasporangium, ovule, the embryo sac. The gynoecium represent female reproductive parts of a flower. The gynoecium may consist of a single pistil, monocarpellary, or maybe more than one, multicarpellary. When there are more than one, the pistil is maybe fused to form syncarpus or maybe free epicarpus. Each pistil has three parts, that is stigma, style, ovary. The stigma serves the landing platform for pollen grain. The style is elongated, long, slender beneath the stigma. The basal bulged parts of the pistil is ovary. Inside the ovary is ovarian cavity, locules. The placenta is located inside the ovarian cavity. Recall the definition and the types of placentation that you have studied in class 11th. Arising from the placenta are megasporangia, commonly called as ovules. The number of ovules in an ovary may be one, wheat, mango, wheat, paddy, mango, of maybe many, papaya, watermelon, orchids. The megasporangium. Let us familiar with ourselves with the structures of a typical angiosperm ovule. Ovule is small structure attached to the placenta by means of stalk, funicle. The body of an ovule is fuses with the funicle region called as hilum. The hilum represents the junction between the ovules and the funicle. Each ovule has one or two protective envelope called integuments. Integument encircles the nucellus except the tip of the small opening called as micropyle is organized. Opposite a micropyle end is a chalacel represent the basal parts of the ovules. Enclosing within the integument is a inner mass of cells called as nucellus. The cells of the nucellus has abundant reserve food materials. Located in the nucellus, embryo sac or the female gametophytes. Ovule generally has a single embryo sac formed from a embryospores. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called as megasporogenesis. The ovules generally differentiate single megaspore mother cells in the micropylar regions of the nucellus. It is a large cells containing dense cytoplasm with a prominent nucleus. A MMC undergoes meiotic division. Microspore mother cells undergoes division of mitotic division. What is the importance of MMC undergoing meiosis? The meiosis results in production of 4 megaspores. The female gametophyte, it is a majority of the flowering plants. One of the megaspore is functional while the other 3 degenerates. Only the functional megaspores develop into female gametophytes. Embryo sac. This method of embryo sac formation forms a single megaspore is termed has monosporic development. What will happen to the ploidy of the cells in the nucleus? The MMC, that is a functional megaspore, and the female gametophytes. Let us study the formation of embryo sac in details. The nucleus of the functional megaspore divides mitotically to form 2 nuclei which move to opposite poles, forming a 2-nucleate embryo sac. The 2 or more sequential mitotic nuclear division results in formation of 4 nucleate and later the 8-nucleate stage, the embryo sac. It is interesting to note that the mitotic division are strictly free nuclear. What is nuclear divisions that are not following immediate by cell wall formations? After the 8-nucleate stage, the cell wall laid down, leading to the organization of typical female gametophytes or embryo sac. Observe the distribution of cell inside embryo sac. The 6 of the 8 nuclei are surrounded by cell wall and organized into cell. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the larger central cell. There is a characteristic distribution of cells within the embryo sac, and the 3 cells are grouped together at a micropylar end that constitute the egg apparatus. Egg apparatus is in turn consists of synergid and 1 egg cell. The synergids have a special cellular thickening at the micropylar tip called filiform apparatus, which plays an important role in the guiding the pollen tubes into synergids. 3 cells are chalacels in and are called as antipodal. The central larger cells mentioned earlier are 2 polar nuclei. A typical angiosperm embryo sac's maturity at 8 nucleated 7 celled. Pollination. In the preceding sections, you have learned to male and the female gametophytes in the flowering plants produced in the pollen grains and the embryo sac respectively. As both the gametes are non-motile, they may be brought together by fertilizations to occur. What is this? How is this achieved? Pollination is a mechanism to achieve this objective. A transfer of pollen grain shed from the anther to the stigma of a style is called pollinations. The flowering plants have evolved amazing area of adaptation to achieve pollination. They take up use of external agents to achieve pollination. Can you think of the list of possible external agents? Kinds of pollination. Depending on the source of pollen, pollination can be divided into three types. Autogamy. It is in this the pollination is achieved within the same flower. Transfer of pollen grain from anther to stigma of a same flower. A normal flower, the open and exposed anther and the stigma. Complete autogamy is rather rare. Autogamy of a flower requires a synchronym in the pollen release and the stigma receptivity. The anther and the stigma should lie close to each other, so that self-pollination can occur. Some plants such as viola, camelina, oxalis produce two types of flower. The chasmogamous flower, which is similar to the flower of another species exposed anther, and the stigma. Cleistogamous flower, which do not open at all. In such flower, anther and stigma lie close to each other. When anther dehiscens in the flower bud, the pollen grain comes in contact with the stigma to effect pollination. Thus, cleistogamous flower is invariably autogamous flower, as in there is no chance of cross-pollinations landing on the stigma. The cleistogamous flower produces a short seed set even in the absence of pollinations. Do you think the cleistogamy is advantageous or disadvantageous to plants? Why? Getinogamy. Transfer of pollen grain from anther to stigma of another flower of same plant. And although autogenic getinogamy is functionally cross-pollination involving a pollinating agents genetically similar to autogamy, similar pollen grains come from the same plant. Xenogamy, transfer of pollen grain from the anther to stigma of a different flower. This is the only type of pollination which during the pollination bring about genetically different types of pollen grain to the stigma. Agent of pollination. The plants use biotic wind, water and one biotic animal agent to achieve pollination. Majority of plants use biotic agents for pollination. Only a small portion of plants use biotic. Pollen grains coming in contact with the stigma is a chance factor in both the wind and water pollination. To compensate this uncertainty, the associated loss of pollen grain, the flower produces enormous amount of pollen when compared to the number of ovules available for the pollinations. The pollination by the wind is more commonly amongst the biotic pollination. Wind pollination also requires pollen grain which are light and non-sticky, and they can be transported in the wind current. They often possess a well-exposed stamen so that the pollen are easily dispersed into the wind. The large feathery stigma can easily trap airborne pollen grain. The wind pollination flower often have a single ovules in each ovary. Numerous flowers packed into the inflorescence. A familiar example is corn cob. In the ear, you'll see a nothing but the stigma style which wave in the wind to the trap pollen grain. The wind pollination is quite common in the grasses. Pollination by water is quite rare in the flowering plants and is limited to 30 genera, mostly monocotyledon. As against this, as you recall that the water is to regulate the mode of transport of male gametes along the lower plants groups such as algae, bryophytes, pteridophytes. It is believed, particularly some bryophytes and pteridophytes, that their distribution is limited because they need water for the transport of male gametes fertilizations. And example of water pollination is Vallisneria hydrilla, which do not grow in the fresh water, and several marine grassy grasses, Zostra. Not all aquatic plants use water for the pollination. In majority of aquatic plants, such that water hyacinth, water lily, the flower emerges above the level of the water and are pollinated by insect and the winds, as in most of the plants. In Vallisneria, the female flower reaches the surface of the water by a long stalks, and the male flower of the pollen grains are released onto the surface of the water. They are carried passively by the water current. Some of them are eventually reach the flowering female flower, the stigma. In another group of water pollinated flowers such as seagrass, female flower remains submerged in water. Pollen sacs are released inside the water. The pollen grains in much of the species are ribbon-like and they are carried passively inside the water. Some of them reach the stigma, achieve pollination. Most of the water pollinated species, pollen grains are protected with a wetting by the mucilage covering. Both wind and water pollinated flowers are not very colorful, do not produce nectar. Why? What would be the reason for this? Majority of the flowering plant uses a range of animals as a pollinating agent. Bees, butterflies, flies, beetles, wasps, ants, moth, and birds. Sunbirds and hummingbirds. And bats are common pollinating agents. Among the animals, insects, particularly bees, are dominating biotic pollinating agents. Even large animals such as primates, lemurs, arboreal, tree-dwelling rodent, or even the reptile, junco lizards and garden lizards have been reported as pollinators in some species. Often, flowers are the flower of animal pollinated plants especially adapted to particular species of animals. Majority of the insect pollinated flowers are in color, fragment rich in nectar, and when the flowers are small and the number of flowers are clusters to inflorescence to make them conspicuous, animals are attracted to the flower by a color or the fragrance. The flower pollinated by the flies and the beetles secretes a foul odor to attract these animals. To sustain animal visits, flowers have to be adapted to rewards to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting rewards, and the flowers of the animal comes visit in contact with anther and stigma. The body of an animal get a coating of a pollen grain, which are then generally sticky in animal pollinated flower. When the animals carry pollen on the body comes in contact with stigma, it brings about pollination. In some species, the floral rewards are in providing safe space to lay eggs. An example is the tallest flower, Amorphous, and which is 6 feet in height. A similar relationship exists between a species of moth and the yucca, where both the species moth and the plants cannot complete their life cycle without each other. A moth deposits its egg in the locus of the ovary and the flower. It gets pollinated by the moth. A larvae of the moth comes out of an seeds and starts developing. Why do you observe some flower by flowering? Observe some flower of the flowering plants or other available to you. The cucumber, mango, people, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out the animals visit them and whether they can be pollinated. You must be potentially observing the flower over a few days and at different times of the day. You would try to see whether these are in correlations with the characteristic of a flower to the animal that visits it. Careful observe that any visitor comes in contact with the anther, stigma, and so as only a visitor can bring about pollinations. Many insects consume pollen and nectar without bringing about pollination. Such floral visits are referred to as pollen nectar robbers. You may also not able to identify the pollinator, but you will still surely enjoy your effort. Outbreeding device. Majority of the flowering plants produce a hermaphrodite flower. Pollen grains are likely to come in contact with the stigma of the same flower. A continuous self-pollination results in inbreeding depression. Flowering plants develop many devices to discourage self-pollination, encourage cross-pollination. In some species, pollen releases stigma receptives are not synchronized. Either the pollen release becomes stigma become receptive or stigma become receptive much more before the pollen is released. In some species, anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevents autogamy. The third device to prevent inbreeding is the self-incompatibility. This is a genetic mechanism to prevent self-pollen and from the same flowering of the same plants. After fertilizing the ovules by inhibiting the pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is to production of unisexual flower. In both male and female, in both present on the same plant, that is castor and maize, monoecious. It prevents autogamy but not getinogamy. In several species such as papaya, male and female flowers are present on different plants, and that is the plant that either male or female dioecious. This condition prevents autogamy and getinogamy. Pollen pistil interaction. The pollination does not guarantee the transfer of right types of pollen. The compatible pollens of same species has stigma. Often the pollen of the wrong type, either the other species or from the same plants, It is self-incompatibility. Also lands on the stigma. The pistil has the ability to recognize the pollen, whether it is a right type compatible or the wrong type incompatible. It is of the right type. The pistil accepts the pollen and promotes the post-pollination events that lead to the fertilizations. If the pollen is of wrong type, the pistil rejects the pollen, preventing the pollen germination on the stigma. The pollen tube growth on the style. Ability of the pistil to recognize the pollen followed by its acceptance or rejection results in the continuous dialogue between the pollen grains and the pistil. The dialogue is mediated by chemical components and the pollen interaction with those of the pistil. It is only in the recent year that botanists have been able to identify some of the pollen pistil in component interacting, leading to recognitions followed by accepted sense of rejections. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube, through which one of the germ pores content of the pollen grains move into the pollen tube. Pollen tube grow through the tissues of the stigma, style and reach the ovary. You would recall that in some plants, pollen grains are shed at two cells, condition vegetative and generative cells. In such plant, the generative cell divides and forms two male gametes during the growth of the pollen tube in the stigma. In plants, they which shed pollen in the three cells condition. The pollen tubes carry two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovules through micropyle, then enters one of the synergy through filiform apparatus. Many recently studied shows that filiform apparatus represent at the micropylar parts of the synergids guides the entry of pollen tubes. All these events from the pollen tube positions to stigma until pollen tube enters the ovules together refers to as pollen pistil interactions. At one point of early, the pollen pistil interaction is a dynamic process involving a pollen recognitions followed by promotion inhibition of pollen. The knowledge of gained in this area would help to a plant breeder to manipulating the pollen pistil interactions even in the incomplete pollinations to get the desired hybrids. As you can easily study pollen germination by distinguishing some pollen from the flowering of pea, chick plant, Clotoradia, Blossom, Viola, Vinca, on a grass slide containing a drop of sugar solutions. After 10 to 30 minutes, observe the slides under the low micropower microscope. You will be likely to see the pollen tube coming out of the pollen grains. A breeder is interested in crossing different species, other genera, to combine a desired character to produce commercial superior variety. Artificial hybridization is one of the major approaches to carbon improvement program. In such crossing experiment, it is important to make sure that only desired pollen grains are used for the pollinations and the stigma is protected from the contaminant from unwanted pollen. This is achieved by emasculations and bagging technique. In the female parent bears a bisexual flower, removal of anther from the flower bud becomes anther dehiscence. Using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flower have to be covered with a bag of suitable size, generally made up of butter, to prevent the contaminant on its stigma with unwanted pollen. This process is called as bagging. When the stigma is bagged, flower attain the receptivity. Mature pollen grains collect from the anther to the male parent, dusted on the stigma. Flowers are rebagged and the fruits allowed to develop. In the female parent produces unisexual flower, there is no need for the emasculation. The female flower bud bagged before the flower opens. When the stigma becomes receptive, pollination is carried out using a desired pollen and the flower is rebagged. Double fertilization. After entering one of the synergid, pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gametes move towards the egg cell and fuse with the nucleus, thus completing the syngamy. This results in the formation of diploid cells, that's the zygote. The other male gametes moves towards the two polar nuclei located in the central cell, fuses with them, produce triploid primary endosperm nucleus. As the involves a fusion of three haploid nuclei, it is called as triple fusion. Since two type of fusion, syngamy triple fusion, takes place in embryo sac, the phenomena is termed as double fertilization, an event unique to the flowering plant. The central cells also also called as triple fusion, becomes primary endosperm cells that develop into a endosperm while the zygotes develop into embryo. The post-fertilization events and structures. The double fertilization event of endosperm, embryo development, maturation of ovules into seeds, ovary into fruits are collectively called as post-fertilization events. Endosperm. Endosperm develop preceding the embryo development. Why? The primary endosperm cell divide, repeated and forms a triploid endosperm tissues. The cells of these tissues are filled with the reserve food materials, are used in the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear division, giving rise to free nuclei. The stage of endosperm development into free nuclear endosperm. Subsequently, the cell wall formation occurs and becomes cellular. The number of free nuclei formation before cellularization varies greatly. The coconut water in the tender coconut the familiar is nothing but the free nuclear endosperm that make of thousands of nuclei. Surrounding the white kernel is a cellular endosperm. Endosperm may be either completely consumed by the developing embryo, groundnut beans, or before the seeds mature, or it may be present in the mature seeds, castor and coconut, be used up during the seed germination. The split open after some seeds, castor, pea, bean and groundnut, fruits and coconut, and look for the endosperm each case. Find out whether the endosperm is persistent in cereals, wheat, rice and maize. Embryo develop in the at the micropylar end of the embryo sac where the zygote is situated. Most of the zygote divides not only after certain amounts of endosperm is formed. This is adaptations to provide assured nutrition to developing embryo. Though the seed differ greatly in the early stages of embryo development, embryogeny is similar in both monocotyledon and dicotyledon. Depicted in the stages of embryogeny, the dicotyledonous embryo, the zygote give rise to the proembryo and subsequently the globular heart-shaped mature embryo. The typical dicotyledonous embryo consists of embryonal axis to cotyledon. The portions of the embryonal axis above the level of cotyledon is epicotyle, which terminates with the pupule or the stem tip. The cylindrical portions below the level of cotyledon is hypocotyle, that is terminates its lower end to the radicle and root tip. The root tip covered with a root cap. Embryo monocotyledon possess only one cotyledon. The grass family, the cotyledon is called as scutellum, which is situated towards one side of the lateral of the embryonal axis. At its lower end, the embryonal axis has a radial and a root cap enclosing an undifferentiated sheath called as coleorhiza. The portion of embryonal axis above the level of the attachment of scutellum is the epicotyle. Epicotyle has short apex and a leaf primordium encloses a hollow follicular structure called coleoptile. Soak a few seeds in the water and say wheat or maize or pea or chickpeas or groundnut overnight, and then split the seeds and observe the various parts of the embryo and the seed. In angiosperm, the seed is a final product of sexual reproductions. It is often described as fertilized ovules. Seeds are formed inside the fruits, and seeds typically consist of seed coats, cotyledon, embryonal axis. The cotyledon of the embryo are similar structure, generally thick and solid due to storage of foods reserved. As legumes, the mature seeds may be non-albuminous, exalbuminous. The non-albuminous seeds may have no residual endosperm as it is completely consumed during embryo development. The pea, groundnut, albuminous seeds retain a part of the endosperm and it is not completely used during the embryonic development. Wheat, maize, barley, castor. Occasionally, in some of the seeds, such as a black pepper, thus beet, the remnants of the nucellus are present, and these persistent nucellus is called as perisperm. The integuments of the ovules harden, has a tough protective coat. The micropyla remain has a small pore in the seed coat. This facilitates the entry of oxygen and water into the seed during germination. As the seed mature, its water content is reduced and seeds become more relatively dry, 10 to 15% of moisture by the mass. The general metabolism activity of the embryo slows down. The embryo may enter a state of dormancy. In favorable condition, the available adequate mitosis moisture, oxygen, suitable temperature. They generate. As the ovule mature into seeds and the ovary develops into fruits, the transformation of ovules into seeds and ovary into fruits proceeds simultaneously. The walls of the ovary develops into wall of the fruit called as pericarp. The fruit may be fleshy guava, orange mango, etc., or may be dry like in groundnut, mustard, etc. Some fruits may be evolve mechanism to dispersal of seeds. You may recall the fruits and dispersal mechanism that you have studied in the early classes. There are relationship between the number of ovules in the ovary and the number of seeds that are present in the fruits. In most plants, by the time the fruit develops, the ovary and other floral part degenerates and fall off. However, in few species such as apple, strawberry, cashew, the thalamus contribute in the fruit formation and called as false fruit. Most fruits, however, develops from the ovary called as true fruits. Although in most of the species, fruits are further results of fertilization, and few species in which a fruit develop without fertilization, such fruits are called as parthenocarpic fruit. Banana is one example. Parthenocarpy is induced by the application of growth hormones. Such plants are endless. Seeds are often several advantages of angiosperm. Firstly, advantages to angiosperm. Firstly, the reproductive process such as pollination, fertilization independent of water, seed formation, and more dependable. Also, seeds have better adapting strategy for the dispersal of a new habitat and helps a species to colonize a rare area. As they may sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protections to the young embryo. Being a product of sexual reproduction, they generate a new genetic recombination leading to variations. Seeds is the basics of agriculture. Dehydration and dormancy of mature seeds are crucial for the storage of seeds, which can be used as food throughout the year and also to raise a crop in the next season. Can you imagine the agriculture in the absence of seeds or in the presence of seeds, which germinate straight away so as to formation of a cannot be stored? How long do the seeds remain alive even after dispersal? The seeds of the large number of species live several years. Some seeds remain alive for hundreds of years. These are several records that old yet viable seeds. The oldest of all is a lupine, Lupines arcticus, excavated from the Arctic tundra, and these seeds germinated and flowered after the estimate of 10,000 years dormancy. A recent records of 2,000 years of old viable seeds is the of the date palm, the Phenoxy dactylifera, 2,000 years discovered during the Anchorage archaeological excavation of King Herodotus' place dead near the Dead Seas. After the completing a brief accounts of sexual reproduction with flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flower plant, asking the following question: how many eggs are present in the embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in the ovary? How many ovaries are present in a typical flower? How many flowers are present in a tree? And so on. Can you think of some flower in which plants contains very large number of seeds, or fruits are one such category that each fruits contain thousands of tiny seeds? Similarly, in case of fruits, some of the parasitic species as Orbolenchus and Striga have been seen. You have seen a tiny seeds of ficus. How many of the trees of ficus develop from the tiny seeds? Many billions of years of seeds does each ficus reproduce. Can you imagine that any other than the example in which tiny structure can produce such a large number of biomass over the year? Apomixis and Polyembryony. Although seeds in general are products of fertilization, a few flowering plants such as some species of Asteraceae grasses have evolved a special mechanisms to produce seeds without fertilization called apomixis. What is a fruit productions without fertilization called? Thus apomixis is a formation of asexual reproduction that are mimic sexual reproductions. These are several layers of developments of apomatic seeds. In some species, the diploid egg cells formed without reduction divisions develops into embryo without fertilization. And so often in many citrus mango varieties, some of the nucellus cells surrounding the embryo sac starts dividing, protrude to the embryo sacs, and develop into embryos. In such species, ovules contains many embryo. Occurrence of more than one embryo in the seed is referred as polyembryony. Take out some of the seeds of orange and squeeze. You will observe the many embryos, different size, shapes from each seeds. Count the number of embryos in each seeds. What would be the genetic variation nature of the apomictic embryo? Can they be called as clones? Hybrid variety of several of our food and vegetable crops have been extensively cultivated. Cultivation of a hybrid tremendous increased productivity. One of the problems that the hybrid is that the hybrid seeds have to be produced every year. If the seeds collected from the hybrids are sown and the plants are polygyny, will do not maintain hybrid character. Production of hybrid seeds is costly. Hence, cost of hybrid seeds becomes more too expensive for farmer. If the hybrids are made into apomictic, there will be no segregation of character in the hybrid progeny. Then the farmers can take upon using the hybrid seeds to raise a new crop year after year. He does not have to buy any hybrid seeds even the next year. Because importance of apomixis in the hybrid seeds industries, active researchers is going on in many laboratories around the world to understand the genetic apomixis to transfer apomictic genes into a hybrid variety. Summary. Flower are a set of sexual reproductions in angiosperm. In flower, androecium consists of a stamen representing the male reproductive organ and the gynoecium consists of a represent female reproductive organ. A typical anther is bilobed dithecous tetrasporangia. The pollen grains develop inside the microsporangia. The four-wall layer, the endodermis, epidermis, middle layer, and the tapetum surrounds the microsporangium. The cells of the sporogenous tissue lie in the center of the microsporangium undergo meiosis, microsporogenesis, to form the tetras of microspore. Individual microspore mature into pollen grain. The pollen grains are represent the male gametophytic generation. The pollen grains have two layered axon intine. They have two cells, vegetative and generative. The three cells are vegetative cells and two male gametes at the time of shedding. The pistil is a three-layer stigma style ovary. Ovules present in the ovary. Ovules is have a stalk called funicles, connective integument, opening called micropyle. Central tissue is the nucellus in which the acrosporangium differentiate. The cells of the acrosporangium, megasporogenous mother cells, divide mitotically. One of the two megaspores form the embryo sacs. Female gametophyte, the mature embryo sac is 7-celled, 8-nucleated. At the micropylar end, the egg is apparatus contains two synergids and egg cells. At the chelacal end is the 3 antipodal. At the center of the egg cell, central cell, is 2 polar nuclei. The pollination is the mechanism of transfer of pollen grain from the anther to stigma. The pollinating agents are further biotic and abiotic. The pollen pistil interaction involves all the events in the landing of pollen grain. The stigma until the pollen tube enters the embryo sac, the pollen is completely compatible or the pollen inhibitions. When the pollen is incompatible, the flowering compatible pollination, pollen grain germinates on the stigma and resulting in pollen tube growth through the style, enter the ovules and finally discouraged to the two male gametes in which one of the synergids and angiosperm exhibits a double fertilization because two fusion events occurred in the embryo sac, namely syngamy and triple fusion. The product of these are the diploid and the triploid endosperm nucellus. In the primary endosperm nucellus, a zygote develops into embryo and the primary endosperm cells forms the endosperm tissues. The formation of endosperms always precedes development of embryo. The development of embryo passes through the different stages such as polyembryony, globular, heart-shaped embryony, and before maturation. Mature dicotyledonous leaf embryo has two cotyledons and one embryonal axis. axis with epicotyl hypocotyl. Embryos are monocotyledons, have single cotyledon. After fertilization, ovary develops into fruits and ovules develop into seeds. A phenomenon called as apomixis found in some of the angiosperm, particularly in the grasses, result in the formation of seeds without fertilization. Apomictic have several advantages in the horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. The phenomenon is called as apomixis. Thank you for watching. Kalank Igniting the Q.